So welcome back from the break. Let's take a look at the concept check question and then let's move on. I only got 2 more sections to do, but the next one might be a little bit longer because it's all about the genetics of speciation. And it's kind of interesting. There's some interesting stuff in here, but let's take a look at the concept check question first. So, based on evidence from previous mass extinctions, what evolutionary and ecological consequences could result if a human-caused 6th mass extinction occurs? Well, first thing is to remember what happens during every mass extinction. A significant number of species goes extinct. And so one of the things that we would expect to notice is a loss of a lot of species. Ironically, that could include ourselves if the environmental change becomes large enough. There's no guarantee that we as a species, just because we are technologically advanced, just because we often use our technology to find a way out of our problem, may necessarily be able to find our way out of this one. It may very well be that we alter the environmental conditions here on the planet significantly enough to lead to our own extinction. That is a very real possibility. Remember, life not only adapts to changes in the environment, it can also cause major changes in the environment that have effects on others. So certainly, if we are causing a 6th mass extinction, we do expect that to play out as a loss of other species, but it could include ourselves. We can't eliminate that as a possibility. But we should also remember that every mass extinction provides opportunities for those organisms that survive. So organisms that survive this potential mass extinction we would expect to be able to take advantage over time of the vacated positions. And a really good example of this in miniature has been the area that was blocked out from human habitation following the Chernobyl event in Belarus, I think it was. It was part of the former Soviet Union anyway, but not within Russia itself. It was in one of the associated regions. That has been blocked off from essentially from human habitation since the event took place, and it is basically returning to nature. And there are organisms that are clearly adapting to the environment, because even though it is highly radioactive by human standards, they are in there surviving and they appear to be doing quite well. This is just in miniature what we might expect to happen. Take humans out of the equation and plants take back the environment and animals come back with them. And we see this happening all the time. So if there is a significant mass extinction event, and if we should happen to be part of that mass extinction, then we would expect life forms to take over. The survivors would take over. What that may look like, no one can predict. And nor would we be wise to try and make a prediction as to what the consequence might be. But we do know that life will survive. That has been borne out by the fossil record many times with the mass extinctions that have occurred over the history of life. It's just that the type of life that is present after the mass extinction is very different to the type of life that existed prior to the extinction. But that is the nature of the extinction. So that's what we would expect to see play out if, in fact, we are now in the midst of the 6th mass extinction that is man-made. Okay, let's look at the next, let's go on to the next section, which looks at the genetics of speciation. So one of the reasons why we can do this is simply because we are now able to take a look at genes in more detail and do so more readily. This is the science of genomics, where we can look at not individual genes, but we can look at whole groups of genes and look at what effect those genes have and do it much more easily in terms of gene expression and gene interaction. So when we start looking at this, what we are looking at is two basic things that go on when we think in terms of genes. We're looking at changes in the timing of gene expression, and we're looking at changes in the location of gene expression. And that gives rise to changes in the spatial appearance, the timing of appearances or development of features, and also the rate at which features appear. All of these are under genetic control. And one of the most interesting ways to look at it these days is a new discipline that's arisen in the last 20 years or thereabouts called evolutionary developmental biology, or EVO-DEVO, as a lot of people like to call it. And this is the fusion of developmental biology, looking at the way in which organisms develop from embryo to adult, and evolutionary biology, the way in which organisms evolve, phylogenetics and the cladistics and those sorts of things that we've talked about previously. It's a fusion of these two disciplines to look at how the development of an embryo reflects the evolutionary history of the organism, to look at how changes at the genetic level give rise to changes at the phenotypic level. And what we've observed over time is that very, very small changes at the genetic level can give rise to substantial changes at the phenotypic level because of the presence of master control genes. And the fact that these genes exist has changed the way in which we think about the role of genes in the development of organisms. But I'll talk a little bit more about that a little bit later in this section. But first, let's take a look at how spatial patterns can change. So the term for looking at the timing or the rate of developmental events during embryological development is called heterochrony. And there are two ideas that are significant in terms of heterochrony. One is called allometric growth. Now, allometric growth can be best demonstrated by looking at the patterns of development in humans and chimpanzees. Why look at those? Because we are extremely closely related. In fact, as you should be aware, chimpanzees are our closest living relatives. We are descended from a common ancestor with chimpanzees that we shared about, or we share about 6.5 million years ago. Some people say about 5 million years ago. Other people say out to about 6.5 million years ago. I think the evidence suggests about 6.5 million years ago. There was a single organism that split into two populations, one of which would become the ancestor of chimpanzees and bonobos, which separated from each other subsequent to the splitting of this species. And the other group would become the ancestor of all of the hominid and hominin species that are related to us as Homo sapiens, so the Australopithecines and the other members of genus Homo. So when we take a look at the way in which chimpanzees and humans develop in utero, in the uterus, there is very little to differentiate a human fetus and a chimpanzee fetus for about the first 3 months of development. And it takes a very skilled embryologist to pick the difference between the two. They are very, very similar, but this is a reflection of the fact that we are so closely related. But then some significant events start to take place in the development of chimpanzees that allow us to differentiate very easily between a chimpanzee fetus and a human fetus. And the most significant is associated with the feet. Now, if you look at a chimpanzee foot, it looks very much like our hand. It has a semi-opposable, I guess we'd have to say big toe, that is semi-opposable to the other toes and allows the foot to be used as a grasping tool. It makes it very difficult for chimpanzees to walk in a bipedal fashion. It doesn't stop them from walking in a bipedal fashion, but it makes it very difficult. The other thing that makes it difficult for them to walk bipedally is the shape of the hips, because the hips cause their legs to splay outwards like that, rather than having them come down and tuck in underwards as ours do, and as any other bipedal organism will. And so when we look for bipedalism in the fossil record, what we're looking for is legs that come down with the knees pushed inwards towards each other so they sit underneath the center of mass. If the legs bow out, then it is almost certain that the individuals were not bipedal, or the organisms were not bipedal. And so these are the two things that we look for in terms of bipedalism, but I think I've mentioned that in a previous part of this section, or part of this presentation as well. But anyway, in the human foot, the big toe stays adjacent to the other digits, which is a juvenile characteristic. Whereas in the chimpanzee, at around 3 months, the big toe starts to migrate away, so it arrives at this sort of position. There are also other changes that go on internally, particularly within the hand and within the foot, that changes the way in which our opposable thumb moves versus the opposable digit for chimpanzees. But we won't go into that. But the big deal here is that it starts to move away. So these are significant changes that are going on and allow us to separate a chimpanzee fetus from a human fetus. But the one thing that doesn't change very much is the shape of the skull. And the reason is quite simple. The skull is the one part of the body that shows the least amount of flexibility during birth. And it's the reason why human birth, at least, is so difficult, because the skull has to make its way out through a very small opening in the pelvis in order to enter the birth canal and for birth to occur. And so that limits the size of the skull for humans. And in fact, a human skull is broken up into 3 pieces, and we have fusion regions in our skull, and where the bones fuse together and lock our skull cap together post-birth. The flexibility there has to occur in part to allow the skull to make its way through the pelvis during birth. Same is somewhat true for a chimpanzee. But post-birth, in a chimpanzee, the skull, or the region that covers the brain, is not going to expand in size relative to the rest of the skull significantly, by virtue of the fact that the chimpanzee's brain is not going to expand very much. It is almost fully formed at birth. It will increase in size a little bit. So there is some room for the skull to increase in size a little bit, but it's not going to increase significantly. Humans, on the other hand, let me move this, are going to show significant increase in the skull volume compared to the rest of the, or the brain volume compared to the rest of the skull. So the human brain case is going to increase significantly in size relative to the rest of the skull. In the chimpanzee, it's going to be the jaw region that increases in size significantly compared to the rest of the skull. In humans, it's the brain case, but that reflects the changes that occur post-birth. In humans, the brain increases in size very dramatically, and it increases in volume compared to a chimpanzee's brain. And so the idea of allometric growth is reflected in the fact that different parts of the skull increase in size relative to the rest of the skull during development. And so in chimpanzees, the allometric growth is reflected in the increase in the jaw, whereas in humans, the allometric growth is reflected in increase in the skull. And so basically, you can think of allometric growth as being differential growth. So differential growth. So remember, this can take place in different parts of the body. Here, we're just using the skull as an exemplar of what's going on. But we should not lose sight of the fact that allometric growth can take place in any body region. So if you want to think in terms of the rest of the body, think about the growth of the skull overall compared to the trunk and the legs. And so if you think in terms of, if you look at a baby and you look at the size of the skull compared to the rest of the body, the skull is relatively large on a baby compared to the trunk and the legs. But if you look at an adult, the trunk and the legs make up a substantial part of the body compared to the skull. Yes, the skull has increased in size quite dramatically, but so is the trunk and the legs, and the size of the trunk and the legs have increased substantially more relative to the whole of the body compared to the skull. That's another example of allometric growth. Different parts of the body increase in size relative to others. So there's a differential growth in different regions of the body. But there is another part that goes along with heterochrony, and that is how the body parts develop with respect to adulthood and sexual maturity. The process is called pedomorphosis and pedogenesis. So pedogenesis is the actual process where juvenile characteristics are retained into adulthood and sexual maturity. Pedomorphosis is the product of pedogenesis. So if we want to think in terms of pedomorphosis and pedogenesis in humans, the retention of the juvenile foot would be a really good example. That's a juvenile characteristic. The fact that the big toe is adjacent to the other digits is a juvenile characteristic compared to the other great apes that we are related to. All other great apes have the semi-opposable big toe on the foot and use it for grasping, but that's a reflection of the fact that they are semi-arboreal, spend a lot of time in trees, whereas we are not arboreal at all. We are ground dwelling. And so retaining the juvenile foot structure is an advantage if you're going to be strictly non-arboreal or terrestrial. But here's another example of pedomorphosis and pedogenesis. So this is a salamander called an axolotl. Let me bring up the pen again. So it's an axolotl. I think that's the correct spelling, axolotl. And it's native to some lakes near Mexico City. And unlike most salamanders, it retains a juvenile characteristic in the or characteristics in the form of gills around the head and a paddle-like tail, because it doesn't undergo a metamorphosis to living on dry land, or at least if it has access to water, it doesn't. And so it retains these juvenile characteristics into adulthood and sexual maturity, and it will reproduce with these juvenile characteristics present. If you want to see axolotls, by the way, if you go to the aquarium that's associated with the Orlando Eye in the tourist district of Orlando, they have an aquarium that has axolotls there, and you can see them, and they're quite unusual looking. And in fact, axolotls caused quite a stir when they were first discovered in Mexico and shipped to Europe, because even though the adults that were shipped to Europe had these juvenile characteristics, when they were allowed to breed in Europe, they changed to terrestrial, the terrestrial form of salamanders. They lost their gills and they lost their paddle-like tail, and they became adapted to terrestrial life. They underwent the metamorphosis to terrestrial life, and that was because they were raised in conditions unlike those that were found in Mexico. And so the retention of these juvenile characteristics is driven by the environment that the adults are raised in. And this is a classic example of pedomorphosis. Retention of juvenile characteristics to adulthood and sexual maturity. So that's another part of heterochrony. So the timing of the development of adult features can be affected. The rate at which different body parts develop can be affected. These all come under the heading of heterochrony. So what we're looking at is changes in the spatial pattern, or what is referred to as the body plan or bow plan, as it has sometimes been referred to. And it turns out that these are under the control of a group of genes that are very, very highly concerned, called homeotic genes. And homeotic genes have been found in a number of different types of organisms. So we know that they are present in a wide range of animals. They've been found in fish, they've been found in Drosophila, they've been found in rats and mice. They are known to be present in fungi because they've been found in yeast. They are known to be present in plants because they've been found in some plants that we use traditionally for plant genetic research. And the common feature of all of these homeotic genes is that they have a highly conserved sequence of amino acids that allows them to bind to DNA. They are DNA binding proteins, and they are regulatory proteins. They serve as transcription factors, and they will bind to specific regions on eukaryotic DNA because they are found in eukaryotes. And as the DNA is bent around, it brings these proteins into alignment with the promoter of a eukaryotic gene. And the range of these homeotic genes and the number of homeotic genes that are involved serve to regulate the expression of eukaryotic genes. But it's a little bit more complicated than that, because it turns out that different eukaryotic genes are regulated by different combinations of these homeotic genes. Now, in animals, the homeotic genes are called Hox genes. So these are homeotic genes that are unique to animals. And our understanding of the role of these homeotic genes has expanded quite dramatically over the last 20 to 25 years. So the way we used to think about the genome of any organism, and particularly we're thinking about this with regards to animals, is that the genome is a blueprint. And if you've ever seen the blueprint of excuse me, of a building or a device, you'll know that the blueprint lays out very specifically and in great detail the shape and location of every individual item within the thing that is being described by the blueprint. It's very, very detailed and very, very specific. And that was the way we used to think about the genome of an organism, that it functioned in that way. And you would need lots and lots of genes to lay out in great detail the specific location and shape and form of every feature of an organism. We don't think that way anymore. And part of the reason why is because of our understanding of the genome. So let me give you a really good example here. If you think in terms of the human genome, we've had that information since the late 90s, what the human genome's genetic composition is. Prior to that, we used to think that humans might have over 1 million genes. That was the original estimation. As we got better at our understanding of the role of genes, the number of genes that humans possessed got lower, 500,000 and then somewhere around 100 to 150,000. By the time the Human Genome Project was being formulated in the mid to late 80s, the number of human genes was estimated to be somewhere between 50 and 100,000. Probably going to need that many genes to lay out a human being. By the time the Human Genome Project was completed at the end of the 1990s, or more or less completed, we understood that humans contained somewhere between 21,000 and 23,000 genes, certainly substantially less than 25,000 genes. And what is more amazing is that Drosophila, the simple little fruit fly, that is the genetics model organism that you learnt about in Biology 1, that only contains somewhere between 15 and 17,000 genes. And if you want an even more spectacular piece of information, a tomato has about 100 times more genes than a human. It takes way more genes to make a tomato than it does to make a human. And it only takes a few thousand less genes to make a Drosophila than it does to make a human. So the question then becomes, why? And the answer is the Hox genes. These are master control genes. These don't control individual genes. These control whole families of genes. And it's because they control whole families of genes that they are so important. And particularly when it comes to animals, because that's where a lot of our understanding comes from, it's the number of master control genes, the Hox genes, and how you can utilize those Hox genes to control all the other genes that becomes important. We now think that the genome, and particularly in the case of animals, that the genome represents a recipe. There's a basic genetic recipe for an animal. We don't know what the total number of genes in that recipe is, but it's not particularly big based on what we've seen compared to other organisms, that are the non-animal organisms. And that recipe is under the control of the Hox genes. So if you want an analogy, think about all the baked goods that you can make, biscuits, pastries, pies, cakes, and the like. There's a basic set of ingredients that you use: flour, liquid, some form of fat, a leavening agent, a rising agent, and so on, sugar and like. And it's the way you mix those together, the ratios in which you combine them, the order in which you combine them, the temperature that you bake them at, and the length of time that you bake them at, that determines what you finally wind up producing. But you start with the same basic set of ingredients. And that's how we're starting to think about this basic genetic ingredient for animals. It's not the ingredients that matter specifically. It's the order in which you mix those ingredients, the ratios in which you mix those ingredients, the way you mix those ingredients, the timing, the length of time you mix those ingredients, and so on, that determines what you wind up with. Basically, you start with this basic recipe for an animal. You get an embryo. We use the term marinate, which is a cooking term, but the embryo marinates in a soup of Hox genes that changes over time. So you'll get different types of Hox genes being expressed in different combinations at different times, in different locations within the body of the animal. And it's the influence of these Hox genes over the basic set of genes in the genome of the organism that gives rise to the body plan, that determines where an arm is going to be located, where a head is going to be located, where a leg is going to be located, the layout of the organs inside the body. All of these features, the basic plan of the organism is under the control of these Hox genes. And you can start to understand why understanding the Hox genes becomes so important, because if they are controlling the layout of the body plan, a small change in the expression of one Hox gene or several Hox genes could have huge consequences to the body plan of the resulting animal. You don't need substantial genetic changes to create substantial phenotypic changes, particularly if those changes occur in these master control genes, the Hox genes. And so that's why we have a real interest in understanding the Hox genes. So let me show you some of the things that we can tell you about the behavior of these Hox genes. So if you recall, I said that the tetrapod body plan originates with fish. And basically all terrestrial tetrapods, no matter where they exist, are just jury-rigged fish bodies. And that's because the basic set of Hox genes that control the layout of a fish body plan are the same set of Hox genes that control the basic tetrapod form of any terrestrial tetrapod. So whether it is an amphibian or a reptile or a mammal, the basic terrestrial tetrapod form originates with the fish body plan, and particularly the idea of a pectoral girdle with pectoral limbs, we call them arms, and a pelvic girdle and pelvic limbs, which we call legs. That basic terrestrial tetrapod originates with the pectoral and pelvic fins of a fish. And the Hox genes that control the basic layout of those limbs are the same ones that are present in fish, and we also find them in all terrestrial tetrapods. But you may then argue that fish don't have fingers and toes. Well, that's true, but that's because we have extra sets of Hox genes that can be utilized in different fashions to control the development of a limb. And so the chicken leg or the chicken wing has been used as an exemplar of what goes on, and we'll learn a little bit more about that when we do the embryology chapter, chapter 47 later in the semester. But here you can see the chicken limb bud, which is the progenitor of the chicken wing, and you have two regions now that determine the basic body plan of a limb. And while you may not realize this, your arm, and I'll try and demonstrate this, this is going to look pretty uncomfortable, but I'll try and do it. Your arm actually shows the same three basic axes that your body has. So you know that your body has a head to tail or anterior to posterior axis. It has a front to back, so ventral to dorsal. Dorsal is back, ventral is front. And then left to right. Those are the three axes that define your body. Well, your arm has those same basic axes, although one of them is called a little bit different. So it does have an anterior to posterior. So the thumb is considered to be anterior. The little finger is posterior. So that's your anterior posterior axis. Dorsal to ventral is back of the hand to front of the hand. So dorsal is back of the hand. Palm is ventral. And then the third axis is not called left to right. It's called proximal, close. So that's the shoulder. And distal or distant, that's the fingers. And to establish the complexity of the limb versus the quote unquote simplicity of a fin simply involves defining not two body axes, but three body axes or three limb axes. And so long as you can define two of the three axes, you automatically define the third. So you don't have to establish the three axes in the limb bud. You only need to establish two. So these two regions establish those two axes. This region establishes the anterior to posterior axis, anterior being here, posterior being here. And this establishes the proximal to distal, so proximal to distal axis. And as soon as you establish two of the three axes, you establish the third one automatically. So dorsal ventral is established automatically by establishing these two axes. But again, it's Hox genes. And it turns out that the Hox gene that establishes the limb pattern or the fin pattern in the zebrafish and contributes to the limb pattern in terrestrial tetrapods is also found in Drosophila. And so the chicken version, the zebrafish version, and the Drosophila version have been interchanged. And the limbs have that are under the control of these interchanged genes develop normally. The introduction of a different version of the gene from another species does not change the normal limb development. This is a reflection of just how highly conserved these genes truly are. And although we would never do this, one could imagine that transferring perhaps the Drosophila or zebrafish gene into a human embryo would not result in any change in normal development either. But that is a presumptive idea, not something that we would ever try to determine experimentally. But given that the interchange of genes that we have interchanged between species has led to normal limb development, we would expect there to be no changes in development. But this is the power of the Hox genes. This is why they're so important. When we look at the body plans of, for instance, a snake, which is this one. So this would be a snake. This is a chicken. The highlighted region here represents the thoracic vertebrae. And the appearance of thoracic vertebrae is under control of a series of Hox genes. The same Hox genes that are giving rise to this small set of thoracic vertebrae in a chicken are the same Hox genes that are being expressed here in the body of the snake and give rise to this large region of the snake that is the elongated body plan. In fact, you could think of a snake as an elongated thorax. Now, other regions of the snake skeleton are also exaggerated, but notice that in terms of cervical vertebrae, these are the cervical or neck. There's a fairly large region of cervical vertebrae reflected in the long neck of an adult chicken, but there are virtually no cervical vertebrae. And in fact, there's no evidence of vertical vertebrae in the snake. And again, this is a reflection of how the Hox genes control the development of the body plan of the animal. And we could also point to the expression of those Hox genes that we talked about in the limb bud. Chickens, of course, are terrestrial tetrapods. They've got limb buds for forelimbs and hind limbs. Snakes are descended from limbed relatives. They are very close relatives to lizards, and in fact, they both belong to the same clade. Snakes and lizards belong to a clade called squamata, and snakes are descended from limbed ancestors, but they have lost the limbs. And not surprisingly, you would expect that the Hox genes that are in control of the limbs are either not expressed or have changed expression in the tissue that would give rise to the limb buds. And we see some vestiges of limbs in some of the quote unquote primitive snakes. These are the constrictor snakes, the boa constrictors and the like, that still have vestiges of the hind limbs. The bones are completely enclosed within the body. They're not connected to a pelvis because there isn't one, but the vestiges of the hind limb bones are still there. A reflection of their descent from limbed ancestors that were shared with lizards. But this is all under the influence of Hox genes. And so it is the subtle change in Hox genes, change in their expression, change in the location of their expression, change in the timing of their expression, changing in the combinations of Hox genes being expressed in different locations that lead to the changes in body plan. And just to reinforce this, the two Hox genes that control the limb development in a chicken are the same two Hox genes that control the limb development in your arm. But here's what is most surprising. The Hox genes that controls the anterior-posterior axis also is involved in brain development. It controls, it is part of the control mechanism for determining the number of neurons that are present in your developing brain. So why can it be involved in that in your brain, but give rise to one of the two defining axes of a limb? Well, that's because in the brain, it's in combination with different Hox genes, and therefore it turns on a different set of genes in nerve tissue versus the genes that are turned on in the limb bud. But see, that's why we can form a very, very complex body plan from a very, very small number of structural genes, because we have a large set of Hox genes that allows us to have more fine control over the structural genes that we've got. If you only have a limited number of Hox genes, there's only so much you can do with the ingredients under your control, the genetic ingredients. If you have a lot more Hox genes, then you have the opportunity then to control those small number of genes in a lot more different types of ways by playing with the combination of Hox genes that control the different structural genes that you've got. And that is what leads to the complexity. In fact, that is the history of the animal kingdom. So when we look at the ancestral animals and we look at all the invertebrates, what we find is that there is a single cluster of Hox genes. And you can see that laid out here. And so there are somewhere in the order of 7 to 9 Hox genes in this cluster. And they are somewhat consequently, or maybe not so consequently, maybe this is deliberate, the order in which they're laid out in the genome is the order in which they are expressed anterior to posterior in the body of the animals. So the first Hox gene in the cluster is the one that is expressed most anteriorly, most closely to the head. And the one that is last in the sequence is the one that is expressed most posteriorly or closest to the tail. And many of the Hox genes in terms of expression actually overlap. So in some regions, it's a single Hox gene, but there is a gradient leading anterior to posterior, and different genes are expressed in different locations, creating gradients of genes and overlaps of genes, which provide positional information to the cells in those regions to tell them where they are anterior to posterior in the animal, which tells them then what type of or what feature they're part of, and then ultimately what cell within that feature they're going to become. You'll learn more about this in when we do chapter 47. When we go from invertebrates to the earliest vertebrates, which are jawless vertebrates, again, more to learn about this in chapter 34, you see a doubling down in the number of Hox genes. There's a duplication of the original cluster of Hox genes to give a second cluster of Hox genes. And this leads to the jawless vertebrates. And so the complexity of the jawless vertebrates is substantially greater than the complexity of the invertebrates, simply because you've got more Hox genes. And that's what allows for a more complex body plan. When you see the evolution of jaws, and you now have jawed vertebrates, you have a second doubling down. So there's a second duplication. So instead of having 2 sets of Hox genes, you now have 4 sets of Hox genes. And this is our situation. We are part of the jawed vertebrates. And so we have a greater diversity of Hox genes, and therefore the capacity to have a greater degree of control over the basic structural genes. And so we create a more complex body plan because we can do more with the basic set of ingredients, the basic set of genetic ingredients. And very subtle changes in the way these Hox genes are expressed can lead to changes in body plans. So here's an example from crustaceans. Arthropods. So Drosophila and Artemia are both extant arthropods, but they share a common ancestor almost 400 million years ago. So there's been a lot of time passed since they last shared a common ancestor. But they do both share a common set of Hox genes, and the Hox gene that's in question here is this one called the UBX gene. This is considered to be Hox gene number 7. So in terms of the region of the embryo where it's expressed, it's a fair ways down the body. And so we find the expression of the UBX gene in the thoracic region of both of these arthropods. But it does different things because the nature of its expression, the nature of what it does is very different in Artemia versus Drosophila. So the original role of UBX was apparently to promote the formation of legs. And that you see in Artemia, sorry. So the expression of UBX in the thoracic region of Artemia promotes the formation of legs. But 400 million years on, when we see it being expressed in Drosophila, remember these two share for a common ancestor 400 million years ago. So in that subsequent time, that since they diverged from their common ancestor, the role of UBX has changed. So the expression of UBX now is taking place in the abdomen, and its expression is suppressing leg formation. So in Artemia, it's in the thorax, or let's say thoracic region. And here it's in the, erase that. Oops. It's in the abdomen. So it's being expressed in two different locations, and it's carrying out two different functions. So as soon as you have the expression of the UBX gene in Artemia versus Drosophila, you have a different role. In Artemia, it promotes leg formation. In Drosophila, it suppresses leg formation. But just a subtle change in the expression of the same gene leads to substantial changes in the body plan. And this is why understanding the role of Hox genes is so important. Another example, this time not in terms of the change in the role of the gene, but a change in the way the gene is regulated. So the example here is the sticklebacks. There are sticklebacks that are marine, and there are sticklebacks that are freshwater. And the freshwater sticklebacks are actually derived from a saltwater ancestor. They became isolated from saltwater and gradually adapted as the saltwater body that they were trapped in became more and more freshwater by the dilution of the salt water. And there are distinct differences between the freshwater sticklebacks versus the saltwater sticklebacks. And the difference is seen in a region behind the pelvic, or the location of the pelvic fins. And the difference is in the formation of a pair of spines in the marine stickleback in a ventral location that are not present in the freshwater stickleback. And the appearance of these spines in the marine stickleback is the product of the expression of the gene called the PITX1 gene. Now, the PITX1 gene is also found in the freshwater stickleback. So the question becomes, is this change in body plan, the presence or absence of these spines in the saltwater stickleback versus the absence of these spines in the freshwater stickleback, a change in the actual gene itself, or a change in the way the gene is expressed? Well, a simple examination of the amino acid sequence of the PITX1 gene product tells us conclusively that it's not a change in the nucleotide sequence. Or if it is, it's a silent mutation because there is no change in the amino acid sequence. So that means there's no change in the protein function. So that can't be the situation. It's not a change in the actual gene itself. But if we look at the gene expression then we do see a difference. So the gene is expressed in the most anterior region, the mouth region of both types of sticklebacks. But it is only expressed in the ventral region where the spines emerge in the marine stickleback. That gene, the PITX1 gene, is no longer expressed in the, excuse me, in the ventral region of the freshwater sticklebacks. And this accounts for the loss of the spines in this region of the freshwater sticklebacks. It's a change in the gene expression. It's the same gene being expressed in two different locations for serving two different purposes. And this again reinforces the idea that your ability to use the Hox genes in a variety of different locations, triggering the expression of a variety of different genes, leads to changes in body plan. And a subtle change in the Hox gene expression in one location can lead to an overall change in the body plan. Now, what's interesting here is that the loss of these spines has led in the lake stickleback also has another phenotypic feature in that there is a vestigial structure in the adult that is the vestige of what would have been those spines if they had developed that are developing in the marine stickleback but failed to develop in the freshwater stickleback. And when researchers looked at the vestigial structures, what they noticed was that there is an asymmetric relationship in those vestigial structures, in that the left-hand vestige is larger than the right-hand vestige. There's this asymmetry. And once this was observed, a number of members of the research team wondered whether this could be observed in mammals that had also shed their hind limb structures, because these spines are functionally equivalent to the hind limb structures, that some mammals underwent as they transitioned from terrestrial habitats to aquatic habitats, because there is a equivalent to the PITX1 gene that is found in mammals, and it is partially responsible for the development of hind limbs. And so it was logical to ask this question. Now, this is not something that you can check very easily, because when you think in terms of those organisms that have shed their hind limbs, the ones that have most definitely shed hind limbs are the cetaceans, the whales and dolphins, and the manatees. The pinnipeds, the seals and the sea lions and so forth, have their front limbs and hind limbs. We call them flippers. And they are derived from these same structures. And so the question then becomes, how do we test this? Well, it turns out that here in Florida, there is a way to test this, because at the Florida Museum of Natural History in Gainesville, it's associated with the University of Florida, there are a number of skeletons of manatees that have been collected over time, unfortunately from manatees that have been killed by collisions with boats and things like that. And so this provided an opportunity for the researchers to at least do some form of check to see if they could see the same phenotypic asymmetry as a consequence of the loss of hind limbs, because manatees still carry vestigial bones for the hind limbs. And it turns out that yes, the left-hand side is a little bit larger than right-hand side. That same asymmetry does exist at the phenotypic level. So this has now prompted the research team to go back and see if they can test this genetically. Now, I have not seen a paper that discusses this. That doesn't mean it doesn't exist. I just haven't come across it. Not that I've been looking particularly hard for it, but certainly it would be interesting to take a look, and perhaps I should take a look. But it's interesting to think in terms of the same sorts of potential genetic changes occurring in the adaptation of terrestrial tetrapods to an aquatic existence, where they shed their hind limbs, and it's essentially the same kind of change in Hox gene expression that led to the loss of the hind limbs as part of that adaptation. I guess more to look at to find out whether this, at the genetic level at least, this is in fact the case. So this is the second last break, because I always have a break at the end of the presentation, as you've, I hope, observed. So this is the last internal break before we cover the last section. So let's take a look at this concept check question, take a quick break, and then I'll see you on the other side with the answer for this. It is likely that Hox genes have played a major role in the evolution of novel morphological forms. Why? See you on the other side of the break with the answer.